So the bacteria had really diverged, and really to degrees that, like I said, seemed unimaginable and would certainly seem unimaginable in the environment we exist in today. In a few of these environments, some light-absorbing proteins developed within some of the bacteria. The first of these were what we call bacteria chlorophyll, followed by chlorophylls A and B. The bacteria chlorophylls were functional in the anaerobic conditions present at the time and produced some excess carbon dioxide pockets along the shoreline. And these carbon dioxide containing pockets of atmosphere, the other chlorophylls, A and B, were able to function and were incorporated into some bacterial cells, which had also incorporated some extra membranes, which had an affinity for the green protein and began to combine water and carbon dioxide in a manner which produced organic compounds through this process of photosynthesis and gave off carbon dioxide, which was no stranger to the atmospheric mix. But the other byproduct, was very unusual and common at the time, was a totally different molecule, free oxygen. That was the beginning of everything. And once it began to be released, the ripple effect was incredibly powerful. At first, reducing characteristics of the atmosphere took the oxygen as quickly as it was produced, but the blue-green bacteria found some excellent microhabitats, which expanded as they increased in number, and as they increased in number, they produced increasingly large amounts of free oxygen. Eventually, there was enough free oxygen available in the majority of locations for the atmosphere to no longer be a reducing one. And with free oxygen available, it became a whole new ballgame. Yes, it was a whole new ballgame once free oxygen became available. New utilization parameters for energy now were possible when new chemical combinations occurred. A chemical called ubiquitin arose, as well as some which were called cytochromes. And they found an association with one another which was energetically useful to each of them, and which, when they associated with some of the membranes and DNA, protein, RNA combinations, formed some bacteria-like cells which utilized oxygen as an electron acceptor. These cells acquired a cell wall by fusion with other anaerobic bacteria so that they became fully aerobic bacteria. As time passed, the aerobic and anaerobic bacteria, both the heterotrophic and autotrophic ones, the ones that got food from the outside of themselves and the ones that made their own food, diversified until every niche in the thermally moderating environment had been occupied. For millennia, these organisms lived and proliferated in their various niches as the environment in many areas moderated even more. However, as some environments moderated, some became more extreme with high salt, high acid, and high temperatures, as well as high pressure. A new form of single-celled organism slowly developed, which was able to occupy these new environments, these more extreme ones. They were similar to bacteria in being without a nucleus or any membrane-bound intracellular structures, though their cell wall material and their translational apparati were slightly different. Ultimately, they moved into all of the extreme environments previously mentioned and began to produce large amounts of methane, among other byproducts. All of these byproducts contributed to the environmental gas mixtures which we see today, with 78% nitrogen, 21% oxygen, with argon and carbon dioxide, and a few other gases making up the rest, though the amount of carbon dioxide is increasing due to the carbon pollution from human endeavors, which we'll address at another time. This new form of prokaryote became known as archaea. In 1977, Rose and Fox published a paper containing just one table illustrating the relationship between the ribosomal RNA fragment cataloged of 13 species. The data revealed three distinct groups, which Rose and Fox described as Eukaryotes, Eubacteria, Archaebacteria, and Eukaryotes. They're named for the presumed ancestor of the eukaryotes, so that's eukaryotes. Though some scientists balked at the idea of three kingdoms replacing conventional two, the three domains of life, archaea, bacteria, and eukarya, eventually became widely accepted. With the more recent availability of vast amounts of genomic data, some now challenge the three-domain tree of life and instead support a tree where bacteria and archaea are the sole domains, with eukaryotes branching from the archaea. But Wose's three domains have not lost their stronghold in biology dogma just yet. Fox Wonders if the archaea would have been missed if a different technique had been employed. Fingerprinting gave a much more black and white outcome than you would have if you were actually using modern sequencing, he says. With the percent identity comparisons that scientists use today, archaea would probably just be considered an odd niche of bacteria. Well, they were when they were first found anyway. They called them archaebacteria. As more time passed, one particular archaean group lost some of its integrity as cell wall and fused with some more of the phospholipid membranes, which are found as remnants of other archaea, which had died, and these surrounded the archaean genetic material with a double layer, and some of these archaea were able to reproduce themselves through an unusual sort of binary process, wherein some proteins had attached themselves to the chromosomes, and copies of these separated after duplicating one another, and the protein spindles, which anchored themselves in other protein, which was later called centrioles, contracted and pulled the chromosomes into opposite sides of the cell, which stretched and underwent a sort of modified fission process, producing two duplicated cells, which had chromosomes surrounded by a double membrane, and which had grown considerably larger in the process of accumulating the spindle apparatus in the double-layered nuclear membrane. These cells were primitive ancestral eukaryotic cells, or eukaryotes, from which the first diplomonads, the most primitive extant group of protozoans, and Chlamydomonas, the most primitive of the Volvocene line of green algae, arose. Once the foundations were laid for the evolution of animal and plant eukaryotic cells, the domain eukarya was off and running, and descended with modification leading to humanity. That's where it's going. It's going strong.